This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of one of humanity's most delicious achievements, the domestication of nuts and cocoa. From ancient forests to modern plantations, these remarkable plants have traveled alongside us for millennia. In this narration, we'll discover how early civilizations transformed wild hazelnuts, almonds, and cacao trees into the cultivated crops we know today. You'll learn about the ingenious methods our ancestors used to select, propagate, and perfect these plants, ultimately shaping the flavors that define our desserts, snacks, and chocolate indulgences. Join us as we trace the fascinating journey from forest floor to kitchen table. Chapter 1. The Upper Amazon Cradle. Deep in the tropical rainforests of what is now southeastern Ecuador, where the eastern slopes of the Andes meet the vast expanse of the Amazon basin, an agricultural revolution began more than 5,000 years ago. Here, in a landscape of extraordinary biodiversity, ancient peoples first brought a wild tree under cultivation, setting in motion a transformation that would eventually gift the world one of its most beloved foods. This tree was Theobroma cacao, whose scientific name translates from Greek as food of the gods, and whose seeds would become the foundation of chocolate. For much of the 20th century, scientists and historians believed they understood the story of cacao's domestication. The prevailing narrative placed its origins firmly in Mesoamerica, the cultural region stretching from central Mexico through Central America. After all, it was in this region that archaeologists had found the earliest convincing evidence of cacao use, dating back some 3,900 years. The ancient Olmec civilization left behind pottery vessels with traces of cacao, and later the Maya and Aztec peoples elevated the plant to sacred status, using it in religious ceremonies and even as currency. Given this rich archaeological record and the cultural significance of cacao in Mesoamerican societies, it seemed logical to conclude that this region was not only where cacao was first used, but also where it was first domesticated. Yet this comfortable certainty began to crumble as plant geneticists examined the living diversity of cacao trees across the Americas. When researchers analyzed the genetic makeup of cacao populations, they discovered something unexpected. The greatest genetic diversity of Theobroma cacao was not in Mesoamerica at all, but rather in the upper Amazon basin of northwestern South America. This finding was significant because in plant evolution, the region with the highest genetic diversity typically indicates the center of origin for a species. Wild cacao trees still grow throughout the Amazon rainforest, displaying a remarkable range of genetic variation that far exceeds what is found among the cultivated populations of Central America. These wild populations show adaptations to different microhabitats within the rainforest, with some thriving in seasonally flooded areas, others in well-drained upland sites, and still others in the transitional zones between forest types. This genetic evidence pointed to a different story, one that would require archaeological proof to be fully accepted. That proof came from an unlikely place, a site called Santa Ana la Florida, or SALF, located in the Zamora Chinchipe province of Ecuador, nestled in the upper Amazon region where the Andes Mountains give way to the vast rainforest below. The site sits at an elevation of approximately 1,000 meters, in a zone where cloud forests meet tropical rainforest, creating an environment of exceptional plant diversity. At this archaeological site, researchers working with remains of the Mayo Chinchipe culture uncovered evidence that would rewrite the history of cacao domestication. The excavations at SALF revealed a complex settlement with elaborate ceremonial architecture, including stone-lined plazas and ritual spaces decorated with carved stonework. The Mayo Chinchipe people who built these structures were clearly not simple foragers, but rather a sophisticated society with specialized crafts, architectural knowledge, and complex social organization. Among the artifacts recovered from the site were numerous ceramic vessels, some utilitarian and others clearly designed for ceremonial purposes. It was from these vessels that the revolutionary evidence of ancient cacao use would emerge. The breakthrough came through 3 independent lines of archaeological evidence, each providing a different window into ancient cacao use. The first involved the painstaking analysis of starch grains recovered from ceramic vessels and stone tools. Starch grains are remarkably durable microscopic structures produced by plants, and they can survive for thousands of years in archaeological contexts. Each plant species produces starch grains with distinctive shapes and surface features, creating a kind of botanical fingerprint. Under high-powered microscopy, these grains can be identified to genus or even species level. When researchers examined residues from the Santa Ana, La Florida site, they identified starch grains with the characteristic ovoid to spherical shape, smooth surface texture, and distinctive hilum pattern of Theobroma cacao. The identification was confirmed through comparison with reference collections of modern cacao starch. The second line of evidence came from biochemical analysis detecting theobromine, a compound that gives chocolate its distinctive stimulant properties. Theobromine is found in high concentrations in cacao, but is rare in other plants, making it an excellent marker for identifying ancient cacao use. Using liquid chromatography mass spectroscopy, scientists detected theobromine residues absorbed into the porous surfaces of ancient pottery. The presence of this compound indicated that these vessels had once held cacao-based preparations. The theobromine signatures were particularly strong in certain vessel types, suggesting specialized uses for different pottery forms in cacao processing and consumption. The third and perhaps most compelling line of evidence emerged from the cutting edge of archaeological science, ancient DNA analysis. DNA molecules can survive for remarkably long periods when protected from environmental degradation, and the organic residues trapped in pottery can preserve genetic material from the plants that were processed in those vessels. By extracting and sequencing ancient DNA from the Santa Ana, La Florida ceramics, researchers were able to identify genetic sequences that matched Theobroma cacao. This molecular evidence provided definitive proof that these ancient vessels had been used to process cacao. The DNA sequences even allowed researchers to determine which genetic group of cacao was being used, revealing connections to populations still found in the Upper Amazon today. What made these findings revolutionary was their age. The archaeological layers from which these samples were recovered dated to approximately 5,300 years ago, pushing back the earliest evidence of cacao use by 1,500 years and relocating its origins from Mesoamerica to South America. This discovery fundamentally challenged the traditional narrative and established the Upper Amazon basin as the oldest known center of cacao domestication. The people responsible for this early domestication were the Mayo Chinchipe culture, a society that flourished in the Upper Amazon during the Middle Holocene period. Archaeological evidence suggests these people had developed complex cultural practices long before the SALF site was occupied. Their pottery tradition shows sophisticated decoration techniques, including incised geometric patterns and applied modeling. The Mayo Chinchipe constructed elaborate stone architecture, including terraces, plazas, and structures that appear to have served ceremonial functions. Carved stone vessels and ornaments demonstrate advanced craftsmanship. The presence of goods from distant regions, including Pacific Coast shells and highland obsidian, indicates that the Mayo Chinchipe participated in extensive trade networks, connecting diverse ecological zones. The Mayo Chinchipe lived in a region of exceptional biodiversity, where the diverse ecosystems of the Andes and Amazon converge. This environmental setting would have exposed them to an extraordinary variety of plant species, including wild Theobroma cacao trees growing in the forest understory. The vertical ecological gradient from high-elevation cloud forests down to lowland tropical rainforest created a compressed zone of incredible plant diversity within a relatively small geographical area. This biodiversity hotspot provided both the raw material and the opportunity for agricultural experimentation. The upper Amazon basin proved to be an ideal cradle for cacao domestication for several interconnected reasons. The region's climate provides the warm temperatures, high humidity, and abundant rainfall that cacao trees require. The rich, well-drained soils formed from Andean sediments offered excellent growing conditions. Theobroma cacao is diploid with a genome size of 430 megabase pairs organized into 10 chromosome pairs. This relatively compact genome, combined with the plant's outcrossing reproductive system, created favorable conditions for maintaining genetic diversity while still responding to selection pressure. Wild cacao trees are native to the shaded understory of tropical rainforests, where they grow beneath the canopy of taller trees. The upper Amazon, with its complex mosaic of forest types, offered diverse microhabitats where different genetic variants of cacao could thrive. This environmental diversity likely contributed to the region maintaining the highest genetic diversity of cacao found anywhere in the world. Modern genetic studies recognize at least 11 genetically distinct populations in the Amazon basin, each adapted to specific ecological conditions. The existence of these distinct genetic lineages, all concentrated in the upper Amazon region, provides powerful evidence that this area served as the long-term evolutionary home of the species. The early uses of cacao in the upper Amazon were likely quite different from how we think of chocolate today. Rather than processing the seeds into solid chocolate, the Mayo Chincha people appear to have been primarily interested in the sweet mucilaginous pulp that surrounds the seeds within the cacao pod. This white pulp has a pleasant, slightly acidic flavor that has been compared to tropical fruits like lychee or mangosteen. When this pulp ferments naturally, it produces an alcoholic beverage with approximately 5.34% alcohol content, and evidence suggests this fermented drink was likely the original attraction of cacao to ancient peoples. The process of making this fermented beverage would have been straightforward for the Mayo Chincha people. After harvesting ripe cacao cods, they would break open the hard outer shell to access the pulp and seeds inside. The pulp-covered seeds could then be placed in containers where natural yeasts would begin the fermentation process. Over several days, these yeasts would consume the sugars in the pulp, producing alcohol and releasing the seeds from their sweet coating. The resulting beverage would have provided both nutrition and mild intoxication, making it suitable for both everyday consumption and ceremonial use. Interestingly, this fermentation process that the Mayo Chinchipe likely employed for making beverages is the same process that remains essential in chocolate production today. During fermentation, complex biochemical reactions occur within the cacao seeds themselves. Enzymes are activated that break down proteins and carbohydrates, while microorganisms modify the chemical composition of the seed. These reactions develop the flavor precursors that will later become the characteristic chocolate taste when the seeds are roasted. Without fermentation, cacao seeds remain bitter and astringent, lacking the appealing flavors we associate with chocolate. The Mayo Chinchipe people, in their pursuit of fermented beverages, inadvertently discovered the crucial first step in creating chocolate's distinctive flavor. The timeline of cacao domestication in the upper Amazon reveals a process that unfolded over millennia rather than centuries. At 5,300 years ago, the Mayo Chinchipe were already using cacao in ways that suggest a degree of familiarity and cultural integration. The presence of cacao residues in both domestic pottery and ceremonial contexts indicates that the plant had already become woven into the social fabric of their society. This level of integration suggests that the initial stages of cultivation and selection may have begun even earlier, perhaps 6,000 or more years ago. The domestication process itself would have been gradual and largely unconscious. Early cultivators would have simply collected seeds from the most productive or flavorful wild trees and planted them near their settlements. Over many generations, this preferential collection would have begun to shift the genetic makeup of cultivated populations. Trees with traits favorable to human use, such as larger pods, more abundant pulp, or better flavor, would have been more likely to have their seeds saved and planted. Meanwhile, traits important for survival in the wild, such as certain chemical defenses or specific seed dispersal strategies adapted to particular animal dispersers, might have become less common in cultivated populations. Some cacao varieties would eventually develop self-pollination capabilities, though this trait would emerge later in the domestication process. What makes the upper Amazon basin particularly remarkable as a center of domestication is that it appears to have served this role not just for cacao, but for numerous other important crop plants as well. The region has been identified as a center of origin and domestication for various fruit trees, including cupuacu, a close relative of cacao in the genus Theobroma, as well as peach palm and various species of inga. This pattern suggests that the upper Amazon was a significant hotspot for agricultural innovation in the ancient Americas, comparable in importance to other famous centers of plant domestication, such as the Fertile Crescent in the Middle East or the highland valleys of Mexico. The discovery of cacao's upper Amazon origins illuminates the sophisticated botanical knowledge possessed by ancient Amazonian peoples. The Mayo Chinchipe and other early cultivators were not simply gathering whatever nature provided. They were actively managing and shaping plant populations through selective cultivation. This required detailed understanding of plant growth requirements, reproduction, and the relationship between seed source and plant characteristics. They needed to recognize which trees produced superior fruits, to understand the timing of harvest and planting, and to provide appropriate growing conditions for young seedlings. The successful domestication of cacao, transforming it from a wild forest tree into a cultivated crop, represents a remarkable achievement of indigenous agricultural innovation that required keen observation and multi-generational learning. Understanding where and when cacao was first cultivated provides crucial insights for modern efforts to conserve and utilize cacao genetic diversity. The wild and semi-wild cacao populations of the upper Amazon represent an irreplaceable reservoir of genetic variation. These populations contain genes that may confer resistance to diseases and pests, tolerance to environmental stresses, or the capacity to produce novel flavor compounds. As climate change and emerging diseases threaten commercial cacao production worldwide, these ancient genetic resources may hold the key to developing resilient new varieties. The convergence of Andean and Amazonian ecosystems in this region created a unique environmental context that fostered innovation in plant cultivation, and the biodiversity it generated remains essential for the crop's future. The story of cacao's domestication in the upper Amazon challenges us to reconsider assumptions about where agricultural innovations occur and how they spread. For too long, the focus on Mesoamerican cacao cultivation overshadowed the primary role of South American peoples in bringing this plant under human cultivation. The new evidence from sites like Santa Ana, La Florida, demonstrates the power of combining multiple scientific approaches, from traditional archaeology to cutting-edge genetic analysis, to reveal previously hidden chapters of human history. These findings remind us that the origins of our most important crop plants often lie in the hands of ancient cultivators whose names we will never know, but whose legacy we enjoy every day. Chapter 2. Cacao's Journey Across the Andes. The dense forests of the upper Amazon basin, where cacao was first brought under cultivation, might seem an unlikely starting point for long-distance trade networks. Yet the archaeological evidence reveals that ancient peoples established remarkably extensive exchange systems that moved cacao and other valuable goods across formidable geographical barriers. Within centuries of its initial domestication, cacao began a journey that would carry it from the shadowed understory of Amazonian rainforests across the towering Andes Mountains to the sun-drenched Pacific coastline. This movement was not a simple migration, but rather a complex process of cultural exchange, selective breeding, and genetic mixing that would fundamentally shape the plant's evolutionary trajectory. When Spanish conquistadors first arrived on the Pacific coast of what is now Ecuador in the early 16th century, they encountered something unexpected. Francisco Pizarro, during his initial exploratory voyage in 1526, observed native peoples cultivating cacao trees in small plantations along the coastal regions. This discovery puzzled early European observers because cacao was known to be a tree of the tropical rainforest, not the drier coastal environments where these plantations thrived. What the Spanish had stumbled upon was the culmination of a multi-millennial process of human-mediated dispersal and adaptation. The story of how cacao reached the Pacific coast and the extensive trade networks that enabled this spread has only recently come to light through a combination of archaeological investigation and advanced genetic analysis. Building on the domestication achievements of the Mayo Chinchipe discussed previously, researchers examining ceramic artifacts from coastal Ecuador found something remarkable: residues from cacao processing in pottery that dated back nearly 4,000 years. Analysis of food residues both adhered to and adsorbed by ceramic walls revealed the chemical and genetic signatures of cacao. These coastal sites, associated with ancient cultures like the Valdivia, Machalilla, and Charrera peoples, showed that cacao had become integrated into Pacific coastal societies by at least 4,000 years ago. The Valdivia culture, which flourished along the Ecuadorian coast from approximately 3,900 to 1,400 years before the present, provides particularly compelling evidence of early cacao use outside the Amazon. Archaeologists working across the region have revealed over 400 archaeological sites, creating an unprecedented window into ancient life. Analysis of ceramic residues from Valdivia locations confirmed what chemical evidence suggested. The Valdivia people were processing and consuming cacao. The presence of theobromine, the distinctive alkaloid compound found in high concentrations in cacao, appeared in vessel after vessel. Ancient DNA extracted from these same ceramics provided definitive confirmation. What makes the Valdivia cacao findings especially intriguing is the timeline they reveal. The cacao evidence from Valdivia sites dates to approximately 4,000 years ago. This places the arrival of cacao on the Pacific coast some 1,300 to 1,400 years after the Mayo Chinchipe were cultivating it in the upper Amazon at 5,300 years ago. This substantial temporal gap tells an important story. The establishment of trade networks capable of moving cacao across the Andes was not instantaneous, but rather developed gradually over more than a millennium. The Ecuadorian Pacific coast and the upper Amazon basin are separated by the imposing barrier of the Andes Mountains, with peaks reaching over 4,000 meters. For cacao to cross this barrier required not just occasional contact, but established routes of communication, strategic alliances, and sustained trade relationships that took centuries to develop and mature. The most compelling evidence for these ancient trade routes comes from genetic analysis of the cacao samples found in coastal archaeological sites. When researchers examined the DNA preserved in ceramic residues from the Valdivia region, they made a startling discovery. The genetic signatures did not match the cacao populations nearest to the coast. Instead, they corresponded to cacao varieties from the Peruvian Amazon, specifically from populations found along rivers like the Nengaritza, Yacuambi, and Zamora, in what is now Zamora Chinchipe province. Genetic markers characteristic of specific Amazonian populations, particularly those from the Marañón and Nanay river systems, appeared in coastal Valdivia ceramics. These rivers flow through the southern part of the Ecuadorian Amazon, hundreds of kilometers from the Pacific coast. This genetic evidence reveals something profound about ancient trade networks. The cacao reaching coastal Ecuador was not simply the result of a gradual, step-by-step diffusion from one neighboring group to another.
Instead, it involved long-distance trade connections that linked specific Amazonian source populations directly with coastal communities. The people of the Valdivia culture were obtaining their cacao from trading partners deep in the Amazon rainforest, maintaining exchange relationships across vast distances and diverse ecological zones. Further evidence of these extensive networks comes from ceramic artifacts found along the Caribbean coast of Colombia. Analysis of residues from ancient pottery in this region revealed cacao genotypes that also originated from the Peruvian Amazon. This finding is remarkable because it indicates that Amazonian cacao was not only moving westward to the Pacific coast, but also northward along complex trade routes that eventually reached the Caribbean coastline. The distances involved are staggering. From the upper Amazon to the Colombian Caribbean represents a journey of well over 1,000 kilometers through some of the most challenging terrain in the Americas. The establishment of these trade routes would have required sophisticated social organization and detailed geographical knowledge. Imagine ancient traders departing from Amazonian settlements, their loads carefully packed with cacao pods cushioned against the rigors of travel. They would need to know the mountain passes through the Andes, where sheer cliffs give way to navigable trails that wind through cloud forests and across glacial streams. They needed knowledge of the navigable rivers that could serve as highways through the rainforest, understanding which tributaries led toward the mountains and which dead-ended in impassable swamps. They had to maintain relationships with communities along the routes, places where they could rest, exchange news, and trade goods. The fact that specific Amazonian cacao populations appear repeatedly in distant coastal sites suggests that these were not random, occasional exchanges, but rather established trade relationships maintained over many generations, perhaps centuries.
What motivated ancient peoples to invest the considerable effort required to move cacao across such distances? Part of the answer lies in the plant's unique properties. The fermented cacao beverage produced by the Mayo Chinchipe and other Amazonian peoples had qualities that made it highly valued in ancient societies. It provided both nutrition and mild stimulation, making it suitable for both everyday consumption and ceremonial purposes. Moreover, cacao pods, with their hard shells protecting the seeds and pulp inside, were well suited for long-distance transport. Unlike many perishable tropical fruits, cacao could survive extended journeys of weeks or even months if properly handled. The movement of cacao from the Amazon to the Pacific coast created opportunities for something that would prove crucial for the plant's long-term success: genetic mixing between geographically distant populations. When traders brought cacao seeds from the Peruvian Amazon to coastal Ecuador, they were introducing genetic variants that had evolved under the specific conditions of their rainforest homeland. On the coast, these plants encountered different growing conditions, including different rainfall patterns with pronounced dry seasons, distinct soil types, and altered temperature regimes. Through the process of cultivation and selection in these new environments, coastal farmers began to create cacao populations with novel combinations of genes. Amazon cacao populations show remarkable diversity, with multiple distinct genetic groups, each adapted to specific environments within the basin. When these diverse genetic lineages were brought to the coast and cultivated together, some genetic variants from the Amazonian source populations proved well suited to coastal conditions and were favored by selection. Other traits that had been advantageous in the rainforest understory, such as tolerance for deep shade or adaptations to constantly humid air, might have been less useful on the coast and gradually declined in frequency. Meanwhile, ongoing trade meant that fresh genetic material continued to flow from the Amazon to the coast, providing a steady supply of new variation for coastal farmers to work with. This genetic mixing had profound implications for cacao's adaptability. When humans began moving cacao seeds between these regions and mixing populations that had been geographically separated, they created opportunities for beneficial gene combinations that might never have arisen naturally. Analysis of modern cacao populations reveals the legacy of this ancient mixing. The coastal cacao of Ecuador shows genetic contributions from multiple Amazonian source populations, not just a single ancestral group. This complex ancestry indicates that coastal peoples were not simply receiving cacao from one Amazonian trading partner, but rather from multiple sources over time. They were actively managing their cacao populations, perhaps deliberately crossing plants from different origins to create new varieties with desired characteristics. Other traits that had been advantageous in the rainforest understory, such as tolerance for deep shade or adaptations to constantly humid air, might have been less useful on the coast and gradually declined in frequency. Meanwhile, ongoing trade meant that fresh genetic material continued to flow from the Amazon to the coast, providing a steady supply of new variation for coastal farmers to work with. This genetic mixing had profound implications for cacao's adaptability. When humans began moving cacao seeds between these regions and mixing populations that had been geographically separated, they created opportunities for beneficial gene combinations that might never have arisen naturally. Analysis of modern cacao populations reveals the legacy of this ancient mixing. The coastal cacao of Ecuador shows genetic contributions from multiple Amazonian source populations, not just a single ancestral group. This complex ancestry indicates that coastal peoples were not simply receiving cacao from one Amazonian trading partner, but rather from multiple sources over time. They were actively managing their cacao populations, perhaps deliberately crossing plants from different origins to create new varieties with desired characteristics.
The archaeological record provides additional insights into the scale and complexity of these ancient exchange networks. Researchers analyzing ceramic artifacts from 19 different pre-Columbian cultures spanning from the Amazon through coastal Ecuador and Colombia to Central America found evidence of cacao use in approximately 30% of the samples tested. This remarkably high success rate, finding cacao in nearly 1 of every 3 ancient vessels across such a wide geographical area and diverse cultural groups, demonstrates that cacao had become an important trade good moving along extensive networks of exchange that connected diverse peoples across thousands of kilometers. The 400 archaeological sites that have been studied across the region represent just a fraction of the total settlements that once existed. Yet even this sample reveals the extraordinary scope of ancient cacao trade. The diversity of cultures showing early cacao use is remarkable. Beyond the Valdivia, archaeologists have found cacao residues in artifacts from the Machalila culture, which succeeded the Valdivia on the Ecuadorian coast around 3,600 years ago. The Chorrera culture, which emerged around 3,000 years ago, also shows clear evidence of cacao processing. Later coastal cultures, including the Bahia, Jamacoaque, and La Tolida peoples, all incorporated cacao into their societies. Each of these cultures left behind distinctive pottery styles and developed unique cultural practices, yet they shared a common interest in obtaining and processing cacao.
The movement of cacao was not unidirectional from the Amazon to the coast. Archaeological evidence suggests that the trade routes operated in both directions, with coastal populations sending their own products back to Amazonian communities. Coastal regions had access to marine resources, salt, and certain minerals not found in the interior rainforest. This bidirectional exchange would have further enhanced opportunities for genetic mixing, as successful coastal varieties might have been traded back into the Amazon, bringing with them genetic adaptations to coastal conditions. Over time, this complex pattern of exchange would have created a network of related but genetically distinct cacao populations, each adapted to local conditions but sharing genetic material through ongoing trade. The timing of this genetic mixing is significant. Analysis of ancient DNA and ceramic residues indicates that the exchange of cacao between the Amazon and the Pacific coast was already well established by approximately 4,000 years ago, within 1,400 years of cacao's initial domestication. This relatively early establishment of trade networks means that the genetic mixing and adaptation processes had been operating for millennia before Europeans arrived in the Americas.
The cacao populations that the Spanish encountered on the Pacific coast in the 16th century were the products of thousands of years of cultivation, selection, and genetic exchange. The chronological framework becomes clear. 5,300 years ago, Mayo Principe cultivating in Amazon. 4,000 years ago, cacao reaches Pacific coast through trade networks. 3,600 years ago, Churrera culture expanding cacao use. These milestones chart the gradual but inexorable spread of cacao from its Amazonian homeland. The intensive cultural interactions that facilitated cacao's spread testify to the sophistication of pre-Columbian societies. These were not isolated groups occasionally encountering each other by chance. Rather, they were interconnected communities maintaining active trading relationships across vast distances. The fact that specific genetic variants from particular Amazonian populations appear consistently in coastal Ecuadorian sites suggests that specific trade routes remain stable over long periods, perhaps maintained by kinship networks, strategic alliances, or specialized trading communities who made the dangerous journey repeatedly, building expertise and relationships over generations.
Understanding these ancient trade routes and the genetic mixing they enabled has important implications beyond historical interest. The genetically diverse cacao populations that resulted from this mixing represent invaluable resources for modern agriculture. Each distinct population contains unique genetic variants that evolved or were selected under different environmental conditions. Some populations may carry genes for disease resistance, drought tolerance, or superior flavor that could be crucial for developing improved cacao varieties in the future. The complex history of exchange and mixing means that even geographically distant populations may share some genetic material, creating opportunities to trace beneficial traits across different genetic backgrounds. The coastal peoples who received cacao from Amazonian traders faced the challenge of cultivating a rainforest plant in quite different environmental conditions. The Pacific coast of Ecuador has a marked dry season, unlike the more continuously humid conditions of the Amazon basin. Coastal soils differ in composition from the typically acidic, nutrient-poor soils of the rainforest. Yet, despite these challenges, coastal cultivators successfully established cacao production, suggesting they developed considerable expertise in managing the plant's requirements. They may have created microclimates by planting cacao near water sources or under the shade of other trees, mimicking the understory conditions of its native rainforest habitat. This adaptation required keen observation, experimentation, and the transmission of agricultural knowledge across generations. The success of these early coastal cacao cultivators set the stage for the plant's continued expansion. From the Pacific coast of Ecuador, cacao would eventually spread northward through Central America, though that part of the story would unfold over subsequent millennia. The genetic mixing that occurred as cacao moved from the Amazon to the coast created populations that were pre-adapted to a wider range of environmental conditions than their purely Amazonian ancestors. This enhanced adaptability, forged through 1,300 years of gradual dispersal, trade, and selection, would prove crucial as cacao continued its human-mediated dispersal across the Americas and, eventually, around the tropical world. Chapter 3. The Birth of Distinct Varieties. The ancient trade networks that carried cacao from the Amazon to distant regions created more than just geographical expansion. As cacao seeds traveled to new lands and fell into the hands of different cultivators, the plant began to diverge into distinct forms. In each region where cacao took root, local farmers made choices about which trees to favor, which seeds to save, and which characteristics to emphasize. Over generations and centuries, these accumulated decisions transformed a single wild species into a constellation of cultivated varieties, each bearing the imprint of the cultures that shaped them. Nowhere was this transformation more dramatic than in the emergence of the variety known as Criollo.
The name itself, meaning native or Creole in Spanish, reflects the European colonizers' recognition that this was the cacao they encountered upon arriving in Mesoamerica, distinct from other forms they would later discover. What the Spanish did not know was that Criollo represented the culmination of a domestication process that had been underway for thousands of years before their arrival. Recent genomic research has revealed the remarkable story of Criollo's origins. By sequencing the complete genomes of 200 cacao plants from populations across the Americas, scientists were able to trace the ancestry of this prized variety back to its roots. The analysis revealed that Criollo descended from a population called Curaray, one of the ancient genetic groups native to the Amazon basin near the modern border between Ecuador and Colombia. However, the Criollo that Spanish conquistadors found being cultivated throughout Mesoamerica had undergone a profound transformation from its Amazonian ancestor. What the Spanish did not know was that Criollo represented the culmination of a domestication process that had been underway for thousands of years before their arrival. Recent genomic research has revealed the remarkable story of Criollo's origins. By sequencing the complete genomes of 200 cacao plants from populations across the Americas, scientists were able to trace the ancestry of this prized variety back to its roots. The analysis revealed that Criollo descended from a population called Curaray, one of the ancient genetic groups native to the Amazon basin near the modern border between Ecuador and Colombia. However, the Criollo that Spanish conquistadors found being cultivated throughout Mesoamerica had undergone a profound transformation from its Amazonian ancestor.
The genomic data indicates that Criollo emerged from what researchers describe as a single domestication event. The timing of this transformation, based on detailed genetic analysis, occurred somewhere between 2,500 and 13,800 years ago, with the most likely estimate being approximately 3,600 years before the present. This timeline places Criollo's origins roughly 1,500 years after the initial domestication of cacao in the upper Amazon, suggesting that Criollo represents a secondary domestication, a process where cultivators took already domesticated cacao and transformed it into something distinctly new. The location of this transformation was Central America, where cacao had been introduced from South American sources through the trade routes described earlier. What makes Criollo's domestication particularly striking is its genetic signature. The analysis reveals an extraordinarily narrow genetic base, resulting from a small founding population that underwent intense selection pressure, creating a variety that was highly uniform genetically but possessed characteristics that Mesoamerican cultivators valued tremendously. The process that created Criollo from its Curaray ancestors involved both the random chance of which seeds happened to be carried to Central America and the deliberate choices of generations of farmers. When cacao first arrived in Central America, it would have been represented by a limited number of seeds or seedlings, creating what geneticists call a founder effect. This initial genetic bottleneck meant that the Central American cacao population started with only a fraction of the genetic diversity present in the Amazonian source populations. From this limited foundation, Mesoamerican farmers began selecting trees with specific desirable traits. Mesoamerican cultivators selected Criollo for superior flavor and enhanced stimulant properties. They also selected for pale seed color rather than the darker hues of wild cacao. This preference for lighter seeds likely stemmed from both aesthetic considerations and correlations with flavor, as seed color relates to compounds affecting taste. Some Criollo varieties developed the ability to self-pollinate, allowing individual trees to reproduce without requiring compatible partners nearby. Additional genes showing strong selection signatures include those involved in disease resistance pathways, though the direction of selection on these genes would have consequences not immediately apparent to ancient cultivators.
Despite the challenges inherent in cultivating this specialized variety, Criollo became the dominant form throughout Mesoamerica, valued by the Maya and Aztecs for its superior flavor profile and potent effects in ceremonial beverages.
While Criollo was emerging in Central America through this process of selective cultivation, cacao in other regions was following different evolutionary paths. In the Amazon basin itself, the original center of cacao domestication maintained extraordinary diversity. These forastero populations, as they came to be called by European colonizers, include groups identified by the river systems where they evolved: Marañón, Contamana, Iquitos, Nanay, Purus, and Guyana, among others. The most recently recognized of these genetic groups, called Caqueta, was identified through advanced genomic analysis of cacao populations from Colombia's Caqueta region. This newly identified group shows unique genetic characteristics that distinguishes it from the other major Amazonian lineages, expanding our understanding of the genetic landscape preserved in the Amazon basin. These Amazonian populations maintained higher genetic diversity than the bottlenecked Criollo. In the Amazon, cacao continued to be cultivated across vast areas with large, interconnected populations that retained access to wild genetic diversity. Selection pressure was less intense than in Mesoamerica, allowing these populations to maintain variation for numerous traits. The forastero varieties tend to be more robust and productive than Criollo, adapted to the diverse ecological niches of the Amazon rainforest.
One particular forastero type that would later become significant is called a melanado, named for its melon-shaped pods. This variety, which originated in the lower Amazon basin, particularly in Brazil, differs from both Criollo and the upper Amazon forastero populations. A melanado is thought to have been domesticated during the 18th century, making it one of the most recent major cacao varieties to emerge. Brazilian cultivators developed a melanado through selection in the Atlantic coastal forests, creating a variety adapted to conditions quite different from either the upper Amazon or Central America. Along the Pacific coast of Ecuador, yet another distinctive cacao variety was taking shape. This variety, which would become known as Nacional, emerged from the ancient cacao populations that had been established on the coast through trade with the Amazon. Nacional's origins are complex, involving genetic contributions from multiple Amazonian source populations. Recent research has traced Nacional's wild ancestry to specific populations in what is now Zamora-Chinchipe province of Ecuador. These populations, brought to the coast through ancient trade networks and mixed together over thousands of years, underwent selection under the coastal environment's unique pressures, including pronounced dry seasons and different soil conditions compared to the continuously humid Amazon basin.
What modern genetic analysis reveals, however, is that the Nacional encountered by Europeans and cultivated in Ecuador through recent centuries represents not a pure ancient lineage, but rather a complex hybrid. The modern Nacional variety incorporates genetic contributions from ancient coastal cacao populations mixed with Criollo and Amolinado varieties introduced from Venezuela within the last century. This genetic complexity makes Nacional a more recent creation than its reputation as an ancient Ecuadorian variety might suggest, though its ancestry does trace back through the coastal cacao populations established millennia ago. Despite this complexity, Nacional became renowned for its distinctive floral aroma. This characteristic flavor profile, whether accurately described as the Arriba flavor after the region where it was cultivated, or perhaps named for other reasons that remain historically unclear, made Nacional highly prized in international chocolate markets. The specific volatile compounds responsible for Nacional's unique aroma represent genetic variants selected under the particular conditions of coastal Ecuador. The emergence of these distinct varieties through selective cultivation created a genetic landscape that would shape cacao's future. When European colonizers began establishing cacao plantations in various tropical regions during the 16th and 17th centuries, they drew upon this existing diversity.
Spanish colonizers took Criollo from Mesoamerica to their colonies in the Caribbean and Venezuela. Portuguese colonizers worked with Amolinado in Brazil. The 18th century saw these varieties spread to new regions as European colonial powers established tropical plantations.
A particularly significant event occurred on the island of Trinidad in the late 18th century. Spanish colonizers had established Criollo plantations there, but around 1727, a devastating blast disease swept through the island's cacao groves. This pathogen, likely a fungal disease, decimated the susceptible Criollo trees. In response, cultivators began crossing the surviving Criollo specimens with introduced Forastero varieties, creating a new hybrid population. By the early 19th century, this hybrid had become established as a distinct variety called Trinitario, named for its island of origin. The Trinitario hybrids represent a different kind of variety, created not through long-term selection in a single location, but through the deliberate crossing of distinct genetic groups. These hybrids combine aspects of Criollo's flavor characteristics with elements of Forastero's robustness. The creation of Trinitario demonstrates how the diversity generated through millennia of cultivation in different regions could be recombined to produce new varieties with novel combinations of traits. Understanding the origins of these distinct varieties has practical implications that extend beyond historical interest. Modern cacao breeding programs actively utilize the genetic diversity preserved in different varieties. Research stations in Ecuador, for instance, maintain collections of nacional germplasm, including searches for remnant trees with pure ancient ancestry, to preserve unique flavor genes. In Trinidad, agricultural researchers continue to develop new Trinitario lines by crossing specific Criollo and Forastero parents, attempting to optimize the balance between flavor quality and agricultural productivity. West African cacao industries, which now produce the majority of the world's cacao, predominantly cultivate Amolinado-derived varieties and face ongoing challenges with disease pressure that drive interest in incorporating resistance genes from diverse Amazonian Forastero populations. The process that created these varieties, selective cultivation operating over thousands of years, offers insights into how domestication works as an evolutionary process. Domestication is not simply humans imposing their will on plants. Rather, it is a co-evolutionary process where human selection pressures interact with the plant's existing genetic variation and reproductive biology to produce changes that neither party could have anticipated. The maintenance of high genetic diversity in Amazonian Forastero populations reflects cultivation practices that allowed continued gene flow with wild populations and maintained large population sizes across vast geographical areas. The complex ancestry of Nacional, drawing from multiple Amazonian sources and later incorporating Criollo and Amolinado genetics, reflects the reality of ancient trade networks and the subsequent mixing of different cacao populations through both deliberate breeding and chance hybridization. These varieties, shaped by thousands of years of human cultivation in different environments and cultures, represent a biological archive of ancient agricultural practices and cultural values. The genetic diversity of Amazonian populations demonstrates the importance of large connected cultivation areas for maintaining evolutionary potential. The hybrid nature of Trinitario shows how catastrophe can drive innovation through genetic mixing. Each variety is a living document, carrying in its genome the accumulated impacts of countless human decisions made over millennia, decisions that continue to shape the chocolate we consume today. Chapter 4. Selection's Hidden Costs. Having traced cacao's journey from the Amazon to Mesoamerica, we now examine how ancient selection fundamentally altered the plant and the hidden costs of those changes. When cultivators in Central America began selecting trees with superior flavor, enhanced stimulant properties, and distinctive appearance, they were conducting molecular breeding experiments whose consequences can be read today in the DNA of modern cacao varieties.
The pale seeds and potent beverages that made Criollo cacao valuable to Maya and Aztec societies came with genetic trade-offs that would only become apparent millennia later. The distinctive stimulating properties of cacao arise from theobromine, a methylxanthine compound chemically similar to caffeine. This molecule gives chocolate its characteristic mild euphoria and gentle energy boost. When Maya and Aztec cultivators noticed that beverages from certain trees produced more powerful effects than others, they began preferentially saving seeds from these potent specimens. By consistently choosing the most stimulating trees generation after generation, they unconsciously enriched their populations for genetic variants that increased theobromine synthesis. Modern genomic analysis confirms this ancient selection pressure. Genes involved in methylxanthine metabolism show clear signatures of positive selection in cultivated varieties, with specific variants strongly associated with elevated theobromine content. The selection was not uniform across all cacao populations. Criollo, the variety that dominated Mesoamerica, shows particularly intense signatures of selection for theobromine genes. This pattern makes cultural sense, given how cacao functioned in Maya and Aztec societies. Cacao beverages were consumed during religious rituals, offered to deities, and drunk by rulers and nobility. A more potent, stimulating beverage served these ceremonial purposes better than a mild drink. The molecular mechanisms underlying this cultural preference involved changes in gene expression patterns that upregulated theobromine biosynthesis pathways, concentrating the alkaloid in developing seeds. Selection for flavor compounds extended beyond theobromine to the pigments that give cacao seeds their color. Wild cacao seeds are deep purple, colored by high concentrations of anthocyanins, the same pigments that create red and purple hues in many fruits and flowers. Criollo cacao, by contrast, produces pale, almost white seeds with dramatically reduced anthocyanin levels. This color transformation did not happen by accident. Genomic analysis reveals strong signatures of selection on genes controlling anthocyanin biosynthesis, with specific variants in the anthocyanin pathway associated with reduced pigment production. The preference for pale seeds likely had multiple motivations. Seed color correlates with flavor because anthocyanins and related compounds contribute to astringency and bitterness. By selecting for white seeds, Mesoamerican farmers indirectly selected for reduced astringency, creating beans that produced smoother, less harsh beverages. Cultural factors may have reinforced this preference. In Maya symbolism, white represented purity and the divine realm. Pale cacao seeds may have been considered more appropriate for offerings to gods than the dark purple seeds of wild trees. Whatever the precise motivation, the result was clear: systematic selection against anthocyanin production that fundamentally altered the biochemistry of cultivated cacao seeds. The challenge of selecting for superior flavor extended beyond individual compounds. The characteristic taste of fine chocolate emerges from the complex interaction of hundreds of volatile and non-volatile molecules produced during fermentation and roasting. Many of these flavor precursors are synthesized by the cacao seed during development. Ancient cultivators selecting for overall flavor quality unknowingly selected for particular combinations of genes affecting multiple biosynthetic pathways. The difficulty was that genetic potential could not be tasted directly in seeds. Farmers could only evaluate the final product after processing. Successful flavor selection required both careful fermentation techniques and patience to assess trees over multiple harvests. This intense focus on flavor and stimulant properties came with consequences that would only become apparent later. The founding population from which all Criollo descended consisted of somewhere between 437 and 2,674 individual trees, with the most likely number being approximately 738 trees. When Criollo emerged through selection from this limited number of founder individuals brought to Central America, the resulting genetic bottleneck trapped harmful mutations alongside beneficial traits. Genomic analysis reveals that Criollo carries a significantly higher proportion of deleterious mutations than Amazonian varieties. These harmful variants affect genes involved in disease resistance, stress tolerance, and basic metabolic functions. The mechanism behind this accumulation relates to how selection operates in small populations. In large wild populations, natural selection efficiently removes mildly harmful mutations from the gene pool. However, in the restricted breeding populations maintained by Mesoamerican cultivators, random genetic drift could overwhelm selection's effectiveness. Slightly deleterious variants that would have been eliminated in the wild could persist and even increase in frequency when population sizes remain small. Modern statistical analysis has quantified this fitness cost precisely. As the proportion of Criollo ancestry in a cacao tree increases, its productivity, measured in kilograms of beans per hectare per year, significantly decreases, with the relationship being highly statistically significant.
The situation worsened because beneficial and harmful mutations can be physically linked on the same chromosome. When ancient farmers selected a tree with superior flavor, they selected its entire genome, including both advantageous and disadvantageous variants. If a flavor-improving gene happened to sit near a disease susceptibility gene, both would be inherited together. Breaking these unfavorable genetic linkages required large breeding populations and many generations. But intensive selection for elite traits kept Criollo populations small and genetically uniform. The practical consequences became painfully evident in Criollo's agricultural performance. The variety proved notoriously susceptible to diseases that wild Amazonian cacao resisted easily. Fungal pathogens like black pod, witch's broom, and frosty pod could devastate Criollo plantations. The trees required careful management, optimal growing conditions, and constant vigilance against pests to produce acceptable yields. Even under ideal circumstances, Criollo yielded less than robust varieties, representing direct evidence that the domestication process imposed measurable costs on plant fitness. In stark contrast, the Amazonian cacao populations that never went through Criollo's severe bottleneck maintained remarkable disease resistance and environmental adaptability. These trees grew in large, interconnected populations with continued access to wild genetic diversity. Selection pressure was less intense, allowing retention of variation in genes affecting pathogen defense and stress tolerance. Genomic analysis of Amazonian varieties reveals that genes involved in recognizing and responding to pathogens maintain high levels of variation, with many different functional variants present. This genetic diversity provides a standing reservoir of resistance that can be drawn upon when new diseases emerge. Beyond seed biochemistry, human selection reshaped cacao's physical form. Some varieties developed dwarf growth habits, producing short, compact trees rather than the tall, slender trunks of wild ancestors. This transformation involved changes in genes affecting gibberellin signaling, the plant hormone pathway that controls cell elongation and growth patterns. Dwarf trees made harvest easier and simplified management in cultivated settings. Pod morphology also changed through selection, with farmers favoring varieties producing larger fruits with thinner shells and higher proportions of seed and pulp relative to husk. Pod shape shifted from the elongated, heavily ridged forms common in wild trees toward smoother, more rounded profiles in some cultivated varieties. One particularly significant morphological innovation was the development of self-pollinating varieties. Wild cacao employs a self-incompatibility system that prevents self-fertilization and enforces outcrossing with other trees. This reproductive strategy maintains genetic diversity but requires compatible mating partners nearby. In cultivated settings, especially during initial establishment in new regions, self-incompatibility could be problematic. A superior tree transplanted to a new location would be unable to reproduce without compatible partners. Some varieties evolve the ability to self-pollinate through mutations in genes controlling pollen pistil recognition, allowing individual elite trees to produce viable seeds alone and greatly facilitating the spread of desired varieties.
Almonds provide a clear parallel example of selection against a dangerous trait. Wild almonds contain high concentrations of amygdalin, a compound that breaks down to release cyanide when seeds are crushed. This chemical defense makes wild almonds toxic to humans. Domestication in the Levant or Iran required identifying and propagating rare mutant trees producing sweet, non-toxic seeds. The genetic basis involves mutations in genes controlling amygdalin biosynthesis, with domesticated almonds carrying variants that block synthesis of this toxic compound. Macadamia, one of the few tree crops domesticated in the 20th century, illustrates morphological selection trade-offs. Wild macadamia produces small nuts with extremely hard, thick shells difficult to crack. Domestication involved selecting for larger kernels and thinner shells that commercial cracking equipment could process. However, selection for shell thinness had to be balanced against the need for shells strong enough to protect developing kernels from insect damage. Modern varieties represent a compromise between accessibility and protection. Size increases appear consistently across domesticated nuts. Brazil nuts, cashews, walnuts, pecans, and hazelnuts all show substantial kernel size increases relative to wild ancestors. This selection involved changes in genes controlling resource allocation during seed development, with cultivated varieties diverting more nutrients into kernel growth. Larger seeds often require longer development times and more resources, potentially reducing total seed number. Ancient cultivators navigated trade-offs between individual seed size and overall productivity, typically favoring larger seeds despite some cost to total yield. These accumulated examples reveal general patterns in how human selection reshapes plant genomes. Selection consistently targets genes involved in biosynthesis pathways producing compounds humans value, whether flavor molecules, oils, proteins, or other products. Morphological genes controlling fruit size, seed size, and plant architecture reliably change during domestication. Genes affecting reproductive biology, particularly those controlling self-incompatibility and flowering time, often show selection signatures as cultivators adapt plants to specific environments and management practices. Plant breeders developing new cacao varieties with better disease resistance can draw upon genetic diversity preserved in Amazonian populations, particularly those genes affecting pathogen defense that show little evidence of having been selected during Criollo's development. Conversely, flavor-related genes intensely selected in Criollo could be introduced into disease-resistant backgrounds, potentially creating varieties combining the best of both genetic groups. This is precisely the strategy pursued with Trinitario cacao, the hybrid attempting to capture Criollo's flavor in a more robust genetic package. The challenge facing modern breeders mirrors that confronting ancient cultivators. Selection for one trait inevitably affects others through genetic linkage and pleiotropy, where single genes influence multiple characteristics. Breaking these linkages and unfavorable correlations requires large breeding populations and many generations, just as developing original domesticated varieties required millennia of selection. Modern breeders possess the advantage of genomic tools to track inheritance of specific gene variants, potentially accelerating breeding by allowing selection based on DNA sequences rather than waiting for plants to mature and express characteristics.
Climate change adds urgency to utilizing the full range of genetic diversity in crops like cacao and tree nuts. As temperature and rainfall patterns shift, gene combinations that proved adaptive in the past may no longer be optimal. Wild and semi-wild populations that ancient farmers drew upon for domestication represent reservoirs of variation that may contain adaptations to future conditions. Some Amazonian cacao populations thrive in areas with pronounced dry seasons or unusual soil chemistry. Genetic variants allowing these populations to tolerate such conditions may become increasingly valuable as climate change creates new environmental challenges for commercial production. The story of human selection on cacao and tree nuts demonstrates that domestication was never simple imposition of human will on passive plants. Rather, it was a complex evolutionary dance where human preferences, plant genetics, environmental pressures, and chance mutations all interacted to produce modern crops. The pale seeds of Criollo, the sweet kernels of almonds, the enlarged nuts of domesticated macadamias all represent successful outcomes of this long coevolutionary process. But each success came with costs and trade-offs, visible now in crop genomes as signatures of ancient selection pressures, accumulations of deleterious mutations, and lost genetic diversity. Understanding this complicated legacy is essential for managing these crops sustainably into the future, preserving the genetic resources ancient farmers bequeathed to us while developing new varieties suited to challenges they could never have imagined. The pale seeds of Criollo, the sweet kernels of almonds, the enlarged nuts of domesticated macadamias all represent successful outcomes of this long coevolutionary process. But each success came with costs and trade-offs, visible now in crop genomes as signatures of ancient selection pressures, accumulations of deleterious mutations, and lost genetic diversity. Understanding this complicated legacy is essential for managing these crops sustainably into the future, preserving the genetic resources ancient farmers bequeathed to us while developing new varieties suited to challenges they could never have imagined. The domestication of cacao and tree nuts reveals a profound truth about our relationship with cultivated plants. From the Mayo Chinchipe farmers who first brought wild cacao under cultivation 5,300 years ago, to the ancient peoples who selected sweet almonds from toxic wild ancestors, human agricultural innovation transformed nature's raw materials into the foods we cherish today. Yet these achievements came with hidden costs. Every gain in flavor, productivity, or appearance required genetic trade-offs that we're only now beginning to understand through modern genomics. The wild and semi-wild populations preserved in places like the Amazon basin represent irreplaceable genetic libraries, containing adaptations that may prove essential as climate change and emerging diseases threaten commercial production. The story of domestication reminds us that agriculture is not conquest, but collaboration, a co-evolutionary dance between human needs and plant biology that continues shaping both partners. Thank you for listening.